Hi, welcome to this lecture on environmental adaptation of animals. Now, for the few videos, we will consider on the transportation of gases in the body fluids. So, we have already discussed that we have gills or lungs or whatever mechanism to take the oxygen from the environment and put it in the circulation. But now, how the oxygen is carried to the cells, and how the carbon dioxide is carried from the cells through with this circulation through the lungs or gill set or whatever. Because in some cases we can't just rely on these lungs or gills at all, because in the surrounding there is not freely available oxygen in the form that we are used. For example, when we are fetus. For the mother, we have learned it quite well. So we have oxygen in environment, and that causes that, okay, there's oxygen in the lungs, and it can be dissolved in the blood, and this dissolving can be enhanced by using hemoglobin. Because it's binding there, then it's going more freely because the partial pressure is lower. But it's not there for the fetus. First of all, before it goes in the fetus, it must be unbinded, released from the mother hemoglobin, and then transported for the fetus blood and binded on the fetus hemoglobin. And then, in the circulation of the fetus, it reaches the cell, then it must be unbinded, released from the hemoglobin. And when it's dissolved in the fetus blood, then it can be going with diffusion to the cell. And we have learned that, okay, in all these steps, the partial pressure of oxygen must be lower. Otherwise, the oxygen is not moving in that direction. And of course, how we can increase the oxygen carrying in the circulation is by using these hemoglobins. And these hemoglobins are colorful. They are actually an example of oxygen transport pigment or respiratory pigment, but quite often we are just calling hemoglobin because that's the way we humans do. But quite often also we think that, okay, the color is because there's iron, there's red color. But actually it's the oxygen binding affecting. So if we have oxygen binding, then it's bright red. But if there is no oxygen, it's purple. And it can be determined more quantitatively by measuring the absorbance. If it's oxygenated, if the oxygen is binded, there are two peaks that are on different wavelength than on the deoxygenated hemoglobin. And actually, when it's oxygenated or fully oxygenated, it's still not completely. So it's not giving the electrodes for the iron, because otherwise the iron will turn in the form that it's not anymore accepting oxygen. And not only it's colorful, but it's also increasing this oxygen carrying capacity. So without the hemoglobin, we could only have 4 milliliter of oxygen per liter of blood. And now, with the oxygen, we can have 200 milliliters per liter. So it's a 50-fold more oxygen carrying capacity. But we have to remember that although we usually think that, okay, it's only the hemoglobin, but it actually, we have a family of globins, even in human genome, and in other animals, we don't even always have the hemoglobin at all. In humans, in embryos or fetus, we have several different hemoglobin subtypes, and even in adult, we actually have two hemoglobin types. Usually we talk only on one, because that's the predominant. And actually, in human genome, we have 12 different genes that are coding different members of globin proteins. And what these hemoglobins are doing, well, even in blood, the hemoglobin is also participating on carbon dioxide transportation. And we have hemoglobin not only in the circulation but also in some neurons, especially in substantia nigra. So it's a part of the brain where it's a little bit more darker because of the hemoglobin. And in these, the neurons, it's there to facilitate the diffusion of oxygen into the cells because there's a binding site. And then, as we have learned during the skeletal muscles, we have myoglobin, but we also have neuroglobin. And they are both for oxygen store. So you can have some kind of you can survive also a short period without any oxygen. And actually, the hemoglobin is not essential for all kind of animals. We have hem in vertebrates and in some other animals, and it's quite well characterized that over in the center there is iron. It's binded on 4 nitrogen, and each nitrogen is part of 5 atom forming ring, and these rings are gathered together, so it's a quite constant structure. And then there are some side groups, and when binding the oxygen, it's binding there in the iron. But we have almost identical structure, chlorocurine, that is in some marine annelids. And the only difference is that on the top and on the left-hand side, there are a little bit different side groups. But although there's iron where this oxygen is bending, this bending is causing now green color. Then we have hematochrine in some marine invertebrates. There are actually 2 iron where several oxygen atoms are binding. And now the oxygenating is causing a violet color. And as you can see, now these rings are not binded together. And so which means that, it's more complex structure, something to do with 3-dimensional things, etc. And then, last but not least, we have hemocyanin in molluscs and arthropods like spiders or octopuses. And now there's no iron at all, there is copper that is binding oxygen. And again, there are these 5 atom rings binded with nitrogen, and now binding oxygen in this complex, turning the color blue, and that makes the octopus blood bluish. So you can see that there is a quite large variety of different respiration pigments beside the hemoglobin itself. And the hemoglobin is actually not this molecule. This is just a hem. Then the globin is the protein that is surrounding it. And if we have more variability on the protein structure, then we can have variability on how easily the oxygen is binding there, or how easily the oxygen is released from these molecules. And how this respiratory pigment is then affecting with the oxygen. We can think it as the same way as in the beginning of this course we were having on the enzymes. So we have ligands that are binding on enzymes. This time we just think that the ligands are oxygen molecules. And these enzymes or substrates are the hemoglobin. So some of the oxygen can be there free in the circulation. Some can be binded on the oxygen binding sites, but then some binding sites can be also freely available. And then there's simple equations to describe that, how eagerly they are occupied, etc. But the idea is that, we only need to know that, okay, what is the affinity? So how eager the oxygen is binding there, and what is the amount of this ligand? So what is the amount of oxygen? And this is the reason why, when analyzing that, okay, how eagerly they are binding or etc., they are usually testing it different oxygen concentration. Because if there's enough oxygen, even when it's not, the affinity is low, all the binding sites of the hemoglobin are binded with oxygen anyway. But if there's very little amount of oxygen, then most of the binding sites are unoccupied. But if we are increasing the affinity, they will bind more eagerly. Okay, and like the enzymes, we have now allosteric modulation. So if we are not having oxygen in the binding, but some other molecules, it can be binding in the same oxygen binding site. So it's competing. And we can have more than one binding site for the oxygen in the hemoglobin. So we can have cooperative binding. So actually in the hemoglobin there are several oxygen binding sites. So, and they can be interacting. So when binding one oxygen, that can affect that, okay, how eagerly the second oxygen or the third or fourth oxygen is binding there. So how it works? Okay, here's the Hem that we already discussed. It can be binded in proteins. The protein can be working alone, like in the myoglobin, so it's surrounding it, or it can be working together with others, like over here in the hemoglobin as a tetramer. And we can see it a little bit more careful in the atom structure. Over here with two different blues, we have the alpha globin and beta globin, and there is a groove on each, so there is a binding site or pocket for this Hem molecule. And as you can see, the Hem is practically flat, and then there in the center there is the iron, but because this pocket is there in the surface, quite easily we can understand that, okay, we can have oxygen molecule binding somewhere over there. And then that affects on the overall structure of this protein surrounding it. And when we have the protein is changing the structure, that can affect on the protein structure on the next molecule, and then affecting, okay, how eagerly the next oxygen is binding there, etc. And as well as we learned in the beginning of this course during the enzymes, we have learned that, okay, we have different kind of globins in the fetal phase and as an adult. So in very early in embryonic, we have a very specific globins, but they are lost quite fast during the first few months. And then we are increasing the amount of alpha globin and gamma globin. But during the birth, we are reducing now the gamma globin that is binding more eagerly oxygen. So that's why we can steal the oxygen from the mother. And now it will be changed to beta globin, but actually not completely. There are about 5% of the hemoglobin in adult humans is another form, not this A2B2 form. And in some other animals, like some fish, there might be 80% having one type and 20% another. So there can be several forms at the same time in the circulation. And the hemoglobin is actually quite widely distributed. Not only in the vertebrates, but in several phyla of animals, and even in some plants, they have found hemoglobin. And it's used in both cases. It can be used for inside the cell or in the circulation in several invertebrates. And in some cases, the animals are producing the hemoglobin only when there's not so much oxygen available. Thank you.